We'll start recording. So we're going to continue our discussion of resistance training adaptations. You guys remember up until now, we've covered most of the changes in strength in their time course that are going to occur in response to changes in recruitment and these so-called neural adaptations, okay? What we're going to get into or continue to build upon from last week today is this idea of muscle hypertrophy. So an increase in the protein content, an increase in actin and myosin that are going to lead to an increase in our measured cross-sectional area, and we're basically going to have bigger muscles, okay? So I'm trying to remember, we talked about protein balance last time, I feel like, and I cannot remember if we completely covered kind of what this slide is going to be here. Now, what I want you all to take in and appreciate is that you're not going to have to know all of the little individual things that are going on here. But what I hope that this does begin to illustrate, if it has not become apparent already, is that there are a host of different signals, okay? Things that are going to cause muscles to grow and get bigger that are going to affect that positive protein balance. It's very, very integrated, okay? And so if what we need basically is we need more protein from down here, okay? We need protein in the muscles. Well, we need transcription so that we can make messenger RNA. We need to make that messenger RNA into actual proteins. And then there's going to be some level of modification after we've made the messenger RNA that needs to happen. And then there's some level of post-translational modification that needs to happen. And we're going to take all of these things and insert them basically into existing sarcomeres or existing muscle fibers. This process, these things that start in the nucleus and then are going to take place in the cytoplasm at the level of the ribosome and then kind of post all of that, are going to be signaled to. We need these signals to get into and tell the nucleus to do this and to tell the ribosomes to start doing what we want them to do. So as we work our way backwards then, well, what are the things that will activate the correct signals? And we think that there is something about the mechanical load, something about lifting something heavy, okay, loading the muscles. There is potentially something about changes in hormones that may go on. And this one is an interesting one that we'll spend a little bit of time talking about, that if you've got a normal hormonal response, that may be sufficient. And you only get problems if you either do kind of supraphysiological levels or if you have a lower than normal kind of response. There is likely to be something from activation of the nervous system that's going to be leading to activation through this calcium signaling pathways, and then potentially also something through this metabolic disturbance. And so we're going to talk a little bit about something over here called mTOR. That seems to be one of the primary intracellular signaling hubs, that if we can do all of these things, especially the load and the metabolic disturbance and things, and we can activate mTOR, that's going to be one of the primary things that that's going to signal on downstream. And then there's some other things that are going to look a lot like what happened with aerobic training. It's activation of AMP kinase, activation of PGC1 alpha, and these things that are going to be triggered by a metabolic disturbance. So it's likely some combination of all of these factors together that are going to drive this increase in protein synthesis. And so the problem that we have in understanding this, especially in intact human muscle, is we can't do a very good job of doing a reductionist model of studying these things, okay? And what I mean by that is I can't give Megan something and block specifically the function of mTOR in her muscles. If I did that, okay, mTOR has a target. It's a, it stands for the mammalian target of an antibiotic called rapamycin. If I gave Megan rapamycin, I can block the effects of mTOR. The problem is that has these very, very wide-ranging additional effects, okay? And so if humans don't have functional mTOR, then you're not going to make it, like past being an embryo, and those kinds of things, okay? If I gave you something to block the function of insulin, that would be a problem. If I gave you more insulin, that could cause problems. If I give you more testosterone, that can be a problem, okay? If I block something to do with calcium, again, there's all of these things that work together. And we can isolate the individual signal and the individual contribution of these things in our cell culture models and in some of our like animal models, especially our mouse and rat models, where we can have these transgenic mice and these knockout mice that have these things or don't have them or overexpress certain of these things. We can do that, but we can't really do this in humans. And so it makes it difficult for us to fully understand exactly which one of these things plays the single most important role, or if you get 2 of the 3, is that going to be enough, or 4 of the 5 or something. Okay, so does that make sense? Because what I'm going to show you guys as we go forward, I'm going to show you some evidence from cell culture models and evidence from some animal models and things as to where we have gotten this idea that all of these things and some of these things down here are important. But it's really hard for us to measure all of this in people. It's hard for us to do a controlled study where we manipulate only one of these little things in people to see what its independent contribution is. Okay, so that's why there's a lot of stuff that we see in animals and then we see in cell cultures that just don't work in people. And I'll tell you some kind of insights and some stories about that as we move along. Okay, so additionally from all of this, as I had mentioned, kind of the big hub for driving this intracellular signaling pathway. So here we have inside the nucleus and things down here. Okay, we're trying to make at a ribosome, we're making things, we've got our messenger RNA, we're making our proteins and stuff here. So we've got stuff that is driving all of this protein synthesis, but we've got insulin and its binding, we've got mechanical loading, we've got something called myostatin that we'll talk about, right? We've got insulin-like growth factor, these androgen receptors that are going to go on, something called Wnt signaling that's going to be right here. You've got all of these different things that are going on. mTOR here, this place, okay, mTOR and mTORC1, it's kind of receptor complexes. These are going to be the hub that are going to drive this stuff with P70S6 kinase that are really, really going to help us. Okay, they're going to activate these things. They're going to help drive initiation of protein synthesis. They're going to block some of the things that block protein synthesis. So if we can get mTOR turned on, we should be able to drive protein synthesis. And there's a lot of stuff upstream of that that's going to play a role in different things. And we've got some other stuff that are going to be involved in atrophy, something about FOXO1, FOXO3, and these things that are also going to be activated. Okay, so very complicated signaling pathways. All right, we know a lot about these things, again, from cell culture and from animal models, but we don't know a whole lot from people. All right, but we'll try to get into a little bit about what this stuff is. I do want you to know a little bit about mTOR. I do want you to know a little bit about P70S6 kinase, because these are sort of the big players in the midst of all of this. Okay, all right, so I asked questions about this on the quiz, and you guys did, for the most part, very, very well. Yes, I know your test grades are not posted. I left the first part of them in my bag, which is at home. The other parts of them are here in my office, and they're graded. I need to put them together so I can calculate your final scores. I should get those up in the morning for you. Anyway, but I did grade you and put your quizzes up. So the myogenic cell cultures, okay? A lot of what we know about what works in the cellular mechanisms of things, these intracellular signals, is going to come from myogenic cell cultures, okay? So much of our research is going to start on this single cell level, okay? The good thing, these cells are all very genetically similar. And so we get a lot of control, right? A lot of experimental control because they're all the same cell. And that gives us control over their external and their internal environment. I can bathe all of the cells in the same stuff. I can apply the exact same number of contractions to them if I want, okay? I can give them X amount of external amino acids and things. I got very, very tight control of these things. One of the issues though, okay, one of the problems with myogenic cell cultures is they are not necessarily indicative of whole muscle. In vivo stands for in an intact animal. So they are not necessarily representative of whole muscle in an animal model, and they are certainly not always representative of whole muscles in people, okay? They lack innervation, which we will see there seems to be something important about initiating the contractions, about having that signal from the nervous system that helps to play some role in things. Okay, we know, okay, there are significantly more factors that lead to muscle development and growth in culture than what seems to work in animals and in humans. So I can do a lot of things in cell culture to augment or inhibit muscle cell growth. But a lot of those things that augment don't augment in an intact animal. But some of the things that inhibit growth don't inhibit in an intact animal. So we start at this particular level, but they're not always going to be indicative. So we do this to find candidate things, and then we work our way up the chain in a lot of instances. Okay. So when I talk about myogenic cell culture, you start with these embryonic progenitor cells. You can look at them. These are going to be cells, if you look on the outside and you stain for things, they're going to be what we call PAX3 and PAX7 positive. And a lot of these myogenic cell cultures, we're just going to pull satellite cells out, okay? And so our embryonic progenitor, basically, if we remove the PAX3, then it becomes a PAX7 positive quiescent satellite cell. We can apply certain things to that satellite cell. As it then goes from being quiescent, where it's just chilling out, okay, we can make it commit to begin to become like these individual myotubes, which are going to be the things that get integrated into the muscle fibers, okay? It's going to start expressing these different sort of myokines. This MIF5 plus something called MyoD, then MyoD, and it's going to have myogenin, and it's going to begin to start expressing something called desmin, which is a structural protein, and alpha actinin, which is also going to be a structural protein. And so what we can see is we apply things to our quiescent satellite cells, and then we check over time, are the cells transitioning basically into these committed satellite cells, into a myoblast, a myocyte, and then several myocytes will oftentimes form together to make myotubes, and the myotubes will then kind of stitch themselves together, okay? You don't have to know what any of these things are. I just want to introduce this in case you're ever looking at a paper or you're at a conference and somebody is talking about these cell cultures, and you're like, oh yeah, I remember Dr. Black talked to us a little bit about those things. I remember what a myoblast is, sort of, okay? I remember that there are cells that are expressing MyoD, and that means we've committed out towards forming something, okay? Okay, so we start off with these cell cultures. In a lot of instances then, we want to check and see then, this stuff is working or seems to play some role in hypertrophy in cell culture. Now let's move it into an animal model. Very often it's going to move into mice, and then it may move up kind of the vertebrate chain from there, okay? The vast majority of our kind of reasonable cellular and molecular signaling data is going to come from animal models, okay? The vast majority of it is going to come from here. And so there's some positive to animal models. You can make them genetically similar or genetically identical. You can very tightly control how much activity you allow them to have. You can very tightly control their feeding, okay? Now we can make some, some people can hand-wring over whether or not you force them into certain feeding and activity patterns, if that's actually indicative of what they would experience, kind of out in the world. It's a very reasonable kind of thing. And if we need to, in these animal models, we can study the whole muscle. So as we talked about last time, I can kill the animal, I can cut out the entire muscle, and then I can do my analysis on all of that if I want to, which is better in some ways than doing one individual muscle cell or taking a single biopsy or something like that. Things that I'm going to call less good, okay? Things that are kind of meh about animal models is that we do not have a great ecologically valid model that relates to humans for inducing mechanical overload, okay? No matter what we try to do, it's hard for us to get animals to do resistance training in a manner that is relatively similar to what people tend to do, okay? So just imagine any of you that have a dog or a cat that we're going to try to teach your dog or your cat to lift weights, okay? Present some problems, right? I don't really care how smart your dog or your cat are. I'm sure they're very, very smart, okay? But that's the thing. It can get to be a little bit of an issue. Now, couldn't we in like primates and those kinds of things? Can we get primates to do some more kind of ecologically valid things a little bit? Yes. I saw the other day that they have taught these certain kinds of chimpanzees to like how to use money, and in the colony, the chimps like they've taught them like the value of certain things by like we give you a certain number of these colored discs, and then you can use those to like buy food that you like and things, and then they pick up on like when they change how many discs it takes to get a certain treat that they want. And also the chimps began to like play games and gamble with themselves. So I'm always astounded by like the intelligence of these other animals and those kinds of things. And maybe that also tells us that gambling is just something that is inherent in all of our brains. Anyway, so the way that we try to model resistance training or mechanical overload in a lot of these animal models is going to be one of these three ways. Okay. The first one is something that we call a chronic stretch model. And I'm going to tell you guys now on the front end, okay? If you are like me and you get a little squeamish about this stuff, that's perfectly okay. But I just want to kind of prepare you that what we do in a chronic stretch model is they go in and they will cut the tendon on a muscle in these animals. They'll cut the tendon loose, and then they'll stretch the muscle out and reattach the tendon so that the muscle is stretched at this long length. So imagine I'm going to cut the tendon on your bicep, and I'm going to stretch it farther down towards your wrist to attach it down there. And so now you're walking around with a chronically stretched bicep or a chronically stretched hamstring or something like that. Okay? They fix the muscle at this longer length that leads to greater stretch, greater overload. Some weird things happen when we apply this chronic stretch. It's kind of interesting, but it's also makes me a little squeamish. I'm calling it synergist inactivation, but what it really is this idea that let's just say I'm going to go in and I'm going to cut 3 of your 4 knee extensor muscles. Okay, I'm going to cut your lateralis, your intermedius, and your rectus femoris, and I'm just going to leave you with your vastus medialis to use in your everyday activities. And we're going to watch then to see what kind of walking around and kind of normal things do as we overload that one remaining knee extensor muscle. So we inactivate the synergist. It's sometimes called synergist ablation, but we either denervate by cutting the nerves to them, or they just go in and they cut the muscle. Okay. So now you're doing normal activities with much, much smaller muscle mass, big overload on those things. And then in some instances, they have been able to train them to do something that approximates resistance exercise. They build little contractions, and the mouse will like whack it with their paw. And when they press on it, right, it has a little resistance to it. And they press the thing and it gives them a treat. They press it again and they get a treat. And then tomorrow they're going to press harder to get the same treat. Okay, so we can teach them to do that. In other instances, they just knock them out and anesthetize them every couple of days, and then they do e-stim on their muscles and things like that. So I have a buddy that as part of his postdoc, they were studying nerve injuries, and they had this rat model of doing these things, and they had to, they basically put this headgear on a rat and ran all of these needle EMG electrodes from the brain down to the muscle. And then they would cut the nerve, right? They would usually cut the perineal nerve, or what we would think of as the tibial nerve, so that the rat couldn't use it. It basically couldn't plantar flex when its back legs on one side. And they'd put it on a treadmill and make it try to walk on a treadmill doing that. And then they would see like what was happening with the electrical activity in different muscles and how it was changing and what was happening on its ability to regrow those axons and things. And he had to do all the different surgeries, and they were implanting the electrodes, and it was all pretty gruesome. Not something that I could have done, but we do these kinds of things. Okay. All right. So what about in us? I'm going to show you a little bit of data from us, But the data in us is going to be much more what I would call descriptive in a lot of ways, okay? We know, okay, we know in a lot of ways that if we do resistance training in people, that for most people, your muscles will get bigger, okay? Typically, we do mechanical loading via resistance exercise or some kind of increased level of activity, especially kind of higher load activity that's going to lead to hypertrophy. So, as I told you guys, we know that it works. So why do we care about the why, right? What's the big deal? If we know that if we send Callie to the weight room and she goes 4 days a week and she progressively overloads and she does what we tell her to do and she does it for 6 months, we know there's about a 95% chance that her muscles are going to get bigger, right? And if that's the case, then why does it matter? Why does it matter if it's mTOR that's the important thing, or if it is PGC1 alpha, or if it's calcium signaling from this, or if it's, you know, estrogen or testosterone? Why does any of that matter? Okay. And so in some ways, I would say it's a very valid excuse, right? It's a very valid excuse. My wife does not care about the mechanism. She just wants to know, if I do this, will it work? Okay. But the idea is that if we understand sort of the how and the why, then potentially we can optimize how we train. We can optimize our loading scheme, our number of days of the week, all of the things that we're doing in an effort to get more gains, right? Keldon, we got to get those gains, right? Get bigger increases in muscle mass, or maybe get the same increase but get it without having to go work out as often or as intensely, which can be a really, really big and important thing for athletes. We can kind of make all of this as optimal as possible, okay? So the problem is that in humans, it's hard for us in intact muscle to study the mechanisms, right? So what we're left with is, well, let's see if we vary the load or the intensity, the total volume, so intensity by reps, the number of days per week, the type of contractions, the metabolic stress, which we're going to talk about metabolic stress as blood flow restriction or training to failure, and those kinds of things. Like if we change one of these, or is there some combination of these that are going to allow us to get the biggest increase in muscle size and the biggest increase, therefore, in muscular strength, okay? So, and the thought is that, well, maybe changing these things optimizes the hormone change, or it optimizes the mechanical load that is transmitted along the sarcolemma, which optimizes the activation of mTOR or something like that, okay? Okay, so we're going to start off, okay, mostly looking at stuff in humans, all right? We're going to talk our way through what I think are a couple of the more what we would call seminal studies. These are things that, this is the study that lots of people cite as to why we think certain things work and other things don't. And then I'm going to present some data that I probably, if I had more time, need to go and look and see if there's been another meta-analysis to look at all of this. But I'm going to show you some data on, here's what we know from kind of people looking at a bunch of literature on, here seems to be the optimum set and rep range. Here seems to be the optimum number of days per week range, etc. Okay? All right. So what do we know about the intensity of exercise? So what do we know about load, right? And hypertrophy. So this is from a paper. The first author's last name is Campos. It was performed at Ohio University and in the late 90s, and it was published in the early 2000s. A girl who was a lab mate of mine at the University of Georgia. I was a PhD student. She was in our lab. She was a master's student. So as an undergrad, she was a participant in this particular study. But what they did was they had 4 groups, okay? They had a control group. They had a group that did high repetitions, low weight, high reps, 20 to 28 repetitions for 2 sets. An intermediate rep range, 9 to 11. Don't ask me why they're using odd numbers. I don't know. It is just as sort of disconcerting to me as it maybe is for some of the rest of you. So 3 sets, 9 to 11 reps, and then low reps. So this is more of a moderate weight, and then lower reps, 4 sets, 3 to 5 reps. So you will note that by doing all of this, they're trying to approximate a similar idea of, or similar-ish idea of what we would call volume load. So this is higher weight, more sets, lower reps. You multiply all of that together and you get some number, and that number is not going to be that different than what we're getting in all of these kinds of things. Okay, we'll talk a little bit about volume in the coming slides and whether that actually matters. Some yes, some no, potentially, but they were doing this. They trained for 8 weeks. They did squat, leg press, and knee extension, and then they went from 2 days a week in the first 4 weeks to then 3 days a week in the last 4 weeks. They then took muscle biopsies from these people. So again, human model, but we've got a biopsy, and we're looking at the average individual cross-section of muscle fibers of type 1, 2A, and they're calling them 2B, but they're really 2X's, okay, of our 3 primary muscle fiber types in a biopsy. Now, the biopsy was taken from roughly the exact same location in the vastus lateralis, okay? But remember, it's not exactly the same fibers. There's some limitations in the midst of all of this. So what I want you guys to look at, okay, what I want you to look at is this is the training conditions, okay? Here, what we see from all of this is the percentage of fiber types. So you will see, and you will note that we basically have no changes in the amount of type 1 fibers. So training does not induce any shift towards type 1, okay? What we do see is that with training, as we would expect with resistance training, type 2X fibers are going to decrease and type 2A fibers are going to increase. That's what we see here, okay? So we're shifting our 2X's to 2A's, just like what happens with aerobic training, just like what happens there. Same kind of a shift. Now, in this particular table, this is going to show us the change in muscle size, okay? This is the change in size of these individual, of our individual fibers. And what we see from this one is going to be that when I train using low reps and high weight, I get hypertrophy in all 3 fiber types. When I train with this kind of more moderate or intermediate weight and my intermediate reps, I also get hypertrophy in all 3 fiber types. When I train with a high weight, okay, or a high set, look, I'm going to get it the 3rd time, I promise. High repetitions, lower weight, I get no hypertrophy. I know you guys look at this and you think, right, statistically, right, what are we seeing here? Maybe we're beginning to nod a little bit towards a little bit of hypertrophy, but it's not nearly on the order that we're seeing in our other ones, okay? So this study tells us that light weights, high repetitions, light weights, think, if you will, our girls doing their pink dumbbells, the little 5-pound dumbbells, doing their curls, okay, over in the huff, doing curls, doing some tricep extensions, right? I don't want to get bulky. Well, they're not going to get bulky because they're not going to get any hypertrophy whatsoever from doing all of that, okay? So this was one of kind of the first studies, the one that people cite very often as to that these kind of low and intermediate stuff are going to give us relatively similar gains and similar changes, but doing high reps, low weight does not induce hypertrophy. Now, I will show you some data later on today, maybe not today, maybe on Wednesday or Thursday, that we can do high reps and we can do lighter weight and we can still induce hypertrophy. But we have to do it potentially a little bit differently than how they're doing it, okay? So this is why, in general, most resistance training prescriptions say do 8 to 12 repetitions, right? Somewhere in that range, and you're going to be fine. It's not going to be a big deal. Okay, so what do we know then if we look at all of this about the effects of exercise intensity on hypertrophy? And so what I am showing you guys here is these are data from the quads and the elbow flexors. So biceps and quads. And it's from a review article that, again, it's a little, it's about 12 years old or thereabouts. So we have more data since then, but I like the graphs that they made in the midst of these. So that's why I'm showing this to you. And what I want you to see is look here in the quads. This is the percent increase in cross-sectional area adjusted to the number of days. So it's percent increase in CSA per day in the training study. So what they're trying to do is be able to find a way to compare studies that go 6 weeks or 8 weeks or 12 weeks or 16 weeks to each other. And we will see that there is probably a little bit of an effect of length of time in the training study on things. So they're trying to approximate that. We won't talk about what's going on over here. These very low intensity ones, there's some issues, right? These ones that are showing some kind of reasonable hypertrophy that are taking place are different than this one. This is some blood flow restriction stuff. But look at the rest, right? Megan, I told you we wouldn't have to do regression, but look, here's regression, okay? Right there. Now, it's a pretty reasonable kind of linear relationship showing that the increase in cross-sectional area that we observe per day of training seems to go up as we get closer and closer to 100% of our 1 rep max, okay? So there seems to be. So this is, we can take away from this that higher stuff, 70, 80, 90% of 1 rep max is probably going to be a little bit better than doing 50 or 60%, okay? That's our takeaway. Pretty clear evidence of that in the quads. If we look at it in the elbow flexors, if you squint real hard, maybe you see sort of the same-ish relationship. I don't know what the hell was going on, like why these people were like super duper responders or something. That's difficult for us to kind of explain and understand, but maybe there's also something else that's kind of going on, okay? This stuff is messy. It's confounded by who is the population. It's confounded by how are we actually measuring and quantifying cross-sectional area. Is it with MRI? Is it with CT? Is it with ultrasound? Is it from a muscle biopsy? There are some issues that make all of these comparisons, we'll just say, tricky. But there does appear to be a bit of a dose response, especially in the quads, right? You guys talk about dose responses in the stats class? You guys know what I mean if I say there's a dose response? Keldon, you know what a dose response is?
Yeah, kind of just like the magnitude of a response from like the prescription of the stimulus.
Okay. So if I have a dose response, it's really this idea that if a little is good, a lot is better, right? Dose response means I train a little bit, I get a little bit bigger and a little bit stronger. I train more and I get bigger and stronger. I train a whole bunch and I get even bigger. So that there is this, it's like I take, we'll just use a very simple analogy that I always waver with my undergrads, but I have 1 glass of wine and I don't get a lot. I don't really feel anything. I have 2 glasses of wine and I get a little buzz. I have 4 glasses of wine and I can't talk straight, okay? And that kind of a thing. Alcohol consumption and your level of inebriation or drunkenness is going to function on a dose response. The more you have, the more of an effect you're going to get. This is a textbook dose response, what a dose response would look like, okay? Okay. Now, from the same particular study, let's talk about exercise volume and hypertrophy, okay? So by volume, what we mean, this is looking at basically the total number of repetitions performed per the number of sessions that you're at per week, okay? So does this mean, right, I'm doing 3 days, like everybody's doing 3 days a week or something, which I think most of these data are all from studies that were doing 3 days per week. But what you can see is I'm doing potentially a lower weight and higher reps versus I'm doing maybe lighter weight and a bunch of reps, or may even be doing a heavier weight and a whole bunch of reps per individual session. These are data from typical dynamic lifting. So you're doing concentric up, eccentric down, okay? And then here is stuff where people did training on an isokinetic dynamometer, where we had them do kind of maximal contractions against the lever arm. And you look at this, right? So think again, correlation, regression, very, very typical kind of shotgun blast, not a dose response. As my repetitions go up, who knows? I got the same amount of hypertrophy here, doing 140 reps per session as I was getting in this thing from doing 20 reps per session. So there is no real clear relationship in either of these things, okay? And so the problem with doing this, okay, and this is just one of the things that is inherent with human research, is it's really difficult for us to tease out the independent effects of the number of repetitions, right, from volume and the load and the intensity of the amount of weight that we're lifting in our contractions, okay? It's hard for us to extricate those two things from each other and figure out what is happening, okay? Because if we try to match load, we try to match on load and then just change our rep numbers, then, that doesn't always work out very well. If we try to keep our rep numbers the same but change the load, then our volume is different. It's a fairly tricky way of doing things, okay? So it's just hard for us to know kind of what the best thing to do is. Now,
I have a question about that.
I'm sorry, Alex?
I have a question about that.
Okay. I probably don't have a good answer. So
I don't think it's that complicated of a question, but what do you think would speak against, and this even came up in one of your critical thinking questions, I believe, what would speak against prescribing more or recommending to prescribe more near max exercise for hypertrophy? I guess the reason why I'm asking this is like traditionally, like still with like ACSM and NSCA still like recommends the traditional like 8 to 12 reps, like 60 to 80% for hypertrophy. Yeah. What speaks against recommending or pushing more towards recommendations of near max exercise?
We're going to get to the study that caused that problem in a couple of slides. And I'll tell you kind of why there was some things like that were based on some hormone data that have not been super replicated, but are still if you read the NSCA prescription books, are still in some instances taken as kind of canon. I don't have a good recommendation for that, Alex. I don't. I don't think it's a problem at all to do more kind of heavy lifting. For some populations, they may make the argument that lifting heavy is inherently more dangerous for them. And then sometimes they may argue that if you're doing, like I'm doing 3 sets of 2 reps or 4 sets of 2 reps, they may argue that, well, as you can see from what I'm going to have here, maybe there is some evidence that doing a little bit more from a volume standpoint is better. But there's also some stuff that says that it's not. So it's just kind of a preponderance of the evidence, and people kind of are just like, yeah, do this. A lot of people don't like to lift super heavy. So I think that's perfectly fine, to be 100% honest. Is there probably some level of diminishing returns where you're not going to lift enough, or maybe you can overtrain yourself or injure yourself? Probably. But I think some instances, maybe we don't lift heavy enough, or we don't prescribe enough heavy stuff. Because especially if you want to increase your actual maximal strength, you've got to lift heavy in a lot of ways. And so I think it's important for athletes and things to lift heavy and not just powerlifters and stuff. Probably also important for grandma to lift heavy. So, Alex, are you in my lab?
No.
Okay. I see one of those desks and I'm like, wait, do y'all have those cabinets back there? Look like the cabinets that are in my lab. I'm like, did you like rearrange my lab and are hanging out in my lab? Anyway, no, unimportant, not a big deal. Even though I'm not sure if you're supposed to be here or not. All right, so here's data from a different review article. This is from a different meta-analysis, okay? And what you can see here is this is single versus multiple sets of resistance exercise and their effects on muscular hypertrophy. So what we can see from this is there are training studies where people do one set, okay? 2 or 3 days per week, one set. A lot of that's some older research. They did like one set of 10 reps to failure, 1 day a week and things like that. And doing one single set per session, you will get some hypertrophy, okay? Here's your effect size of about 0.25 standard deviations. So it's still a pretty small effect size. Doing multiple sets, it's a little bit bigger. Maybe it's not quite a moderate effect size, but it's a little bit better. And then if you break out the kind of single versus, then you break the multiple sets out into 2 to 3 sets or 4 to 6 sets, there does seem to potentially be a bit of a dose response when we do all of this, okay? Now, this doesn't necessarily jive with this, okay? There's a little bit of a difference on kind of how they're looking just at sets versus looking at repetitions and volume load and things, and so it complicates the matter. But there is some evidence that maybe if you have the choice, doing more than one set is probably, especially in more advanced folks, is probably going to be a more powerful stimulus than doing one single set, okay? Okay, now we're going to talk about the effects of frequency per week, the number of days per week that you exercise, and the effects on hypertrophy. And so what I want to show you here, which I think highlights one of our problems with this, okay? The honest answer is there's not been a lot of exercise studies, training studies, where we do 1 day per week. There's not been a lot where we do like 5 or 6 days per week. And so almost all of our data comes from doing like 2, 3, or 4 days a week of training. Most of them are 2 or 3 days per week. And so we don't have a lot of data on either end of things to really be able to say, is 1 day going to be the equal of, 2 days or 3 days or something? And we don't have a lot where people are like, yeah, man, we're going to freaking get in there and lift the same muscle 5 days. We're going to squat 5 days per week and just see how terrible we all feel in a month, okay? And so because of that, we don't have a lot of spread in this. And so here, you look at this 2 versus 3 days. We got one study from 4. We got this one like 12 session per week shit that's going on way out here. I really should go and actually pull that study and show the data from that particular study, and we should talk about kind of what all is going on. But you look here, there doesn't seem to be a particularly pronounced effect, but again, we've kind of locked ourselves in. Same deal, this is in the quads. You look in the bicep, and again, they're mostly 2 and 3. If we had some more studies that had done 4, maybe you could argue that we're starting to look a little bit like there could be a dose response, but I don't know. There's probably not enough evidence for us to really talk about all of that. And so again, because most of the recommendations are 2 or 3 days per week, that's what people tend to test. And so we don't have a lot of evidence on either end, okay? So, but in the 2 to 3 day range, there's not a huge amount of things. Okay, now I'm going to show you guys this one, and I think this one was a pretty cleverly done study, despite the fact that the guy that is the first author was a PhD student in the lab that I did my PhD in Georgia while I was a master's student at Georgia. And he may have been the most arrogant jackass that I've ever encountered. He's in the top 2 that I've encountered as a person that was a graduate student. He was really, really insufferable. And I'm glad that he left the lab after, by the time that I got into that particular lab. But he's also a physical therapist. He works at the University of Alabama at Birmingham. And they have a really, really, really fabulous muscle researcher there. His name is Marcus Baumann, and he's Scott Bickles, this guy's name. Hopefully Scott has mellowed with his age, but Scott did some studies with Marcus. This is all stuff out of some stuff with Marcus's lab, where they're very interested in kind of, can we train people? What's the optimum dose? But then especially in older adults, if we get them trained, what can we do from a frequency standpoint to get them to hang on to their strength and hypertrophy gains? So what can we do? Like, what's the minimum thing once you've gotten trained up a little bit? What's the least amount that you can do in an effort to kind of stave off some level of sarcopenia and those things? Okay. So what I'm going to show you guys here, okay, these are data from muscle biopsies. So it is myofibular cross-sectional area, and we've got type 1 fibers and type 2. Oh, I'm sorry. No, I apologize. It is pre, and then it's 16 months. So they did 4 months of training. And I actually believe this is men, and I think it's men and women that we see. This is just average myofibular cross-sectional area. And what you can see is they did 3 days a week, okay? So this panel and this panel are identical here. They did 3 days a week of training, okay? Very traditional kinds of training for 4 months, and you can note that they got about a 50% or so increase in average cross-sectional area. It's a big, big increase, okay? They then, for a further, okay, for a further 16 weeks, and then another 16 weeks. So they basically did for the next 8 months. So this is a whole training program for a year. They did 1 day per week, okay, with lower repetition. So they took what they were doing here, and they did it only 1 day a week at lower reps. So they were only getting a ninth of the stimulus that they were getting from here. And then here, they did 1 day a week where they were doing this, okay? And so what I want you guys to sort of note when they were doing all of this kind of thing is that 1 day per week with lower reps, and they lost a little bit from here, but they were still increased from pre. And then when they were doing the 1/3 volume thing, they were on average. I don't know what the hell was happening in our ladies here, but I believe this is the guys. They actually continued to increase with just 1 day a week. They certainly weren't returning back down here to baseline. Okay, so there's some really fascinating evidence on this of once I can get something out there and going, I can hang on to a lot of it. There's a lot of interesting stuff on atrophy and how people lose things, and it becomes really, really interesting and really important for like space flight and people that are in long-term care facilities that are confined like to bed rest and stuff, which is a whole different kind of situation. But this, I think, gives us a little bit of an idea, especially here, that I went from 3 days to 1 day and I continued to see some stuff. So maybe 1 day a week is going to be sufficient. It may not be sufficient, although you would argue it might not be sufficient in more advanced lifters or younger lifters or things, but maybe it is. Okay, there's a lot of really interesting stuff out there. If anybody is really interested in kind of studying muscle and muscle hypertrophy and sort of sarcopenia and age-related kind of changes and things, going to work with Marcus, he always has multiple postdocs and things in his lab. They've got a shit ton of funding, doing a lot of genetic research and stuff. I'm going to show you guys some more of that a little bit later on. He is a wonderful human and a wonderful scientist. So you should always think about going and doing that. Okay. Now, more confusing data that I want to highlight some stuff on. And I will do this only because I'm going to show you this, and I don't have an answer for it. Okay, I don't have an answer for this. I don't know why. Maybe you guys have an answer. You can tell me. This was given to me as part of my doctoral comprehensive exams, and I still don't know what it means. The professor that gave it to me as a question didn't know what it meant. He said there was no right answer to it. He just wanted to see what the hell I would say. Welcome to being a PhD student. Okay. So we've talked about intensity, we've talked about days per week, we've talked about reps and things. What about contraction type? Okay, does performing concentric contractions or concentric and eccentric contractions or isometric contractions, do those matter? Okay, so we're going to start with this. Now, keep in mind, this is from an animal model. These are in mice. Okay, and what they have done in these things is this IMT, this is isometric training. This S stands for shortening, so this is concentric training. And then this one is lengthening movement training or eccentric only training. So they have 3 groups of mice. They lifted roughly the same load, okay? Isometrically, concentrically, and eccentrically. And they got basically identical amounts of hypertrophy from doing all of this, okay? So one of the things is that it all seems to work here. There's human evidence, as I will show you in the coming slides, that concentric eccentric contractions may in some ways be superior to concentric only, or especially superior to isometric only. But here we have an animal model of doing all of this, and they found that they all work. And so is it related to kind of something, the only thing that's really different is this time under tension, because they were getting less force concentrically. They were doing more contractions to try to match some things. And was it a way they were trying to match stuff? Does this all work in animals, but it doesn't work in people? We don't have a damn clue as far as I know. I don't know anything. I don't know much more. Nobody has ever given me a particularly great explanation for all of this. The authors in the paper like throw their hands up and say, this is not what we thought was going to happen. It's not supported by the human data. We don't know. So, and I can tell you guys, this fellow, Greg Adams, was a postdoc of my PhD mentor. My PhD mentor worked at NASA, and then he went and worked at the University of California at Irvine, in what was arguably one of the 3 or 4 best muscle physiology labs that they've just kind of recently wound it down. Greg retired. The other older guy that he worked with, his name was Ken Baldwin, that he worked with, they kind of wound the lab down and all retired. But they were about as good at this as anybody in the entire world. And they don't have an explanation. I think it's worth sharing with you guys that sometimes even the really bright people find stuff and they always kind of throw your hands up and say, we don't know, okay? That's not my way of telling you to all go push against the wall and do isometric training, or to just get in your squat and hold your squat down at the bottom and be isometric. But maybe, I don't know. I would argue that if isometric really did a bunch of stuff and it really worked like this, then we would see yoga instructors that were just these like giant ripped people, and they are not. We do a lot of isometric stuff in yoga, which is, you know, if you're not very flexible like me and you don't do a lot and you got to hold those things, it can be quite the workout. Okay, how am I doing on time? I get really, really kind of wordy in the midst of all of this. So, so I told you there's some human evidence that eccentrics are magic in humans. And so here you can see this is net, what they call net area under the curve. This is from a group at the University of Texas at Galveston. This is actual measured myofibrillar protein synthesis rates at 4.5 and 8.5 hours post a bout of resistance training. So this is arterial and venous catheters, infusion of radioactive amino acids, and they're looking at the actual turnover rates, synthesis rates in human skeletal muscle via biopsy and those kinds of things, okay? So, and there are some problems with this as well, but what I want you to notice, they did eccentric only training or concentric only training. This is a single session. And what you will note is that here's my resting synthesis rate. Eccentric and concentric both evoke increases in synthesis. You get a bigger effect eccentrically. At 8.5 hours, that has pretty much gone away. But if you look at kind of the net area under the curve over the total time that we've got all of this, you get a larger increase in synthesis rate from the lengthening contractions than you do from the concentric contractions, okay? Now, here we have the same group, okay, but they actually took all of this. This is one session, okay, one session. Remember, to evoke changes in size, we need multiple sessions over multiple days. So yay, it works in one. There's a lot of things that work in one. The synthesis stuff shows a lot of weird things that don't always translate out into kind of what we see in the whole body. And so here, they came back and they trained people, lengthening contractions only, shortening contractions only. And maybe if you squint really, really hard, the eccentric is a little bit better than the concentric. Certainly if you were a bodybuilder, certainly if you were an athlete and you were really trying to maximize muscle mass, this difference here, although it's relatively small, right, Keldon, we could do one of those, what's the weird cheating analysis that I always yell at Aaron about, that the Australian guys promote, where you basically cheat and you talk about practical effects and things. What's that called? Do you remember? No. There's another way that a lot of the kind of sport performance people advocate, Like, is this effect like meaningful? That athletes care a lot about like that last 2% or something, and it's not statistically significant. Anyway, this one, my point was whole body data from MRI measuring cross-sectional area. No differences. Concentric and eccentric were basically the same. And this is after 8 weeks or 10 weeks of training. But if you squint hard, maybe there's a little bit of a difference. Okay. So we see some here in synthesis rates, but we don't really see it here. It doesn't always translate. Okay. Now, here's some additional data. Again, here we've got eccentric training arm, concentric training arm. And again, these are some bad graphs. We've got a group that was training fast and a group that was training slow, concentrically fast, eccentrically fast. Okay. Remember, your concentric arm that trains fast doesn't get very much load. And so you look at the percent change. This is at the proximal, at the middle, and the distal site. I think these were in the biceps. Okay. So proximal here, up by your shoulder. Middle is in the belly of the muscle. And then distal site is going to be down here more towards your elbow. And this is MRI data. And we look at the change in cross-sectional area in the proximal site and the mid site. Proximal, mid, and distal. And in this one, especially when you train fast, the eccentric one evokes hypertrophy because you're getting a lot of force. When you train slowly and you're actually getting more force concentrically, you're going to get some concentric change, but probably greater eccentrically. Here's the changes in their strength that we're going to see, which looks just like our data from the crazy graph, that when you train eccentrically, you get better at going eccentrically. When you train concentrically, you get better at going concentrically. Those things. Okay, so here's some additional data that maybe eccentric is a little better than concentric, especially if there's some speed to it. Okay, so if you've got athletes trying to train and go fast and work on contracting fast, eccentrics can be important. Okay, so what do we take from all of that? What is your take home? Does contraction type matter? I've shown you no data on isometrics because there's not any. People don't normally train isometrically, right? Not a lot of that goes on. Okay, and so there's not much evidence. There's not much evidence that it works. There's just not much evidence at all because people don't. That's just not how we work out. We don't train isometrically. Okay, we had a guy that came looking for a job interview. He actually interviewed in the cycle where we hired Cellular. That was his cardiovascular disease plan. Take people that have high blood pressure, make them train isometrically, and we're going to lower their blood pressure. And he had some preliminary evidence that it was actually working and doing some things. So it was kind of interesting. Obviously not interesting enough because we hired Cellular instead of him in that particular way. So if you ask me, I think there's probably something to the eccentric contractions. So you need to do concentric. If I was to recommend, I would say do concentric, eccentric. There's some additional data. There's some additional human data. There's some stuff from some NASA data, some spaceflight stuff. There's also some data from atrophy studies. When you unload muscles, so like you put somebody in a cast or you put them on bed rest, that the muscle that's going to get casted in a more lengthened position. So you put a cast on my upper arm, okay? My tricep shrinks, my bicep doesn't. You put me in a leg cast and my quad shrinks, my hamstring does not, okay? There's something magic about being in a lengthened position, okay? There's something about stretch while it's being loaded, or at the very least just being stretched, that has some profound, not fully understand or appreciated effects on hypertrophy, or at the very least on preventing atrophy. Okay, and so I'm happy to share. We got a whole bunch more stuff on all of that. We did some unloading stuff and some things when I was a PhD student. I'd be happy to chat or show you guys some papers on all of that if you really care. Okay. Now the stuff that everybody has been all hot to trot and waiting for, I'm sure. And I need to get a different paper and put it in here for you guys to see for next time, okay? What do we know about metabolic stress? What do we know about reaching a particular sort of fatigued metabolic endpoint, okay? An endpoint where maybe there is high levels of lactate, high levels of acidity, right? Depletion of creatine phosphate, high levels of calcium that are out and circulating, okay? Reaching some critical metabolic endpoint, right? So what am I going to show you guys here? These are data from a paper that was published in 2010, and I'm going to show you this to try to talk to you about metabolic endpoints. Then we're going to work backwards to some data that I can remember when we first read this paper when I was a master's student, and the extent to which it totally like fucked our minds up. We thought it was all made up. I still to this day think there's a chance that some of it wasn't made up, but there are some problems. But it's really, really fascinating, okay? And this thing sort of spawned a whole kind of process of doing things differently. Okay, so here in the left panel, what I am showing you is these are people that were doing different kinds of exercise in an MRI unit. They were in the magnet, and what we are measuring here is we're measuring the depletion of creatine phosphate in their muscle. And the idea behind all of this is the more creatine phosphate you deplete, right, the more metabolic stress there is for those particular contractions. And so what you can see here is here we have our control where they're just doing this kind of intermittent, light, low level, intermittent contractions. They get a little bit of creatine phosphate depletion from pre, and then it levels off, okay? This is just kind of very nice and easy contractions. The white open circles here are very, very high force, very heavy contractions, okay? Things where you got to hold something, you're going to do it for 2 minutes, they're going to fatigue very, very much. So you get this very large depletion of creatine phosphate. So much greater fatigue, much greater metabolic stress here versus here, okay? The other ones that we have, we're going to have 20, 30, and 40% of 1 rep max, okay? And those are going to have been performed plus, okay, plus they're applying a blood pressure cuff during this exercise. And then we have, this is a moderate pressure in the cuff, and then we have 20% at a high pressure. So 20% at a high pressure, we're basically completely occluding blood flow to the muscle. And what I want you to note is that statistically, we're getting relatively similar amounts of depletion of creatine phosphate from all of these things as we are during our high force contractions. So what we have done is we are taking this low force contraction, and by either completely or moderately occluding the influx of oxygenated blood to those muscles during exercise, we are going to induce more metabolic stress, and we're going to take a low force contraction and make it fatigue you faster than it normally would and create a similar metabolic stress as what you would get doing high force contractions, okay? Does that make sense? Are you guys with me? Because if you're not, then this next stuff is really going to screw you up, okay? So this is the idea behind what we would call blood flow restrictive training or Katsu training or something. It was originally called Katsu. So here's one of the original papers on this by a fellow, his last name is Takarada. And so what you can see is this is an MRI detected change in cross-sectional area. And they have the biceps brachii, the brachialis, and the triceps brachii. So you've got the arm muscles here, right? You got your 3 arm muscles. We have traditional high load training, 50 to 80% of 1 rep max, okay, kind of traditional high load training. They have low load training, which is going to be 30 to 50% of 1 rep max, and then low load with vascular occlusion. So you're doing curls with the blood pressure cuff on up here so that I lift the low load, but I reach the similar metabolic endpoint as I do lifting my high load. And what you will note basically from all of this is what you can see here is that you're getting, in some instances, you're getting more hypertrophy with the low load with occlusion than you are with the high load by itself. And you're definitely getting more from this versus the low load. So I've now taken and I'm having people lift a light weight, and yet I'm getting equal or greater hypertrophy than I do from lifting a heavy weight. Okay? There are likely some relatively profound implications of all of this, right? You take people that we were talking with Alex, why shouldn't we all lift heavy? Maybe there are people that can't lift heavy, people that are rehabbing from an injury, people that are old and frail, and you're worried you're going to fracture something with them. Okay? So I think all of these things are potentially things we've got to consider. All right? So that's what's going on with this blood flow restricted training. We know a lot more about this now. You can do the occlusion. They've done a lot of research on how much occlusion pressure do you put in there. Do you take the cuffs off between sets and between days, and kind of all of that kind of stuff, like what's going to happen. Okay. All right, let's talk very briefly, and then we'll get out of here for today before we get into some of the more molecular stuff. Okay. How many of you guys have heard the term periodization? Who's heard about periodization? I'm seeing some noddings of head. Alex, in your mind, what does periodization mean?
I mean, there are a lot of different approaches to periodization, but generally, I guess it's like cycling through different phases in terms of using intensities and rep ranges to be able to achieve whatever goal there is at the end. So let's say, for example, like max strength is the goal, be able to periodize certain set and rep ranges leading up to that.
Okay. If you guys had to pick out, that Alex is right. If you had to pick out a different name for periodization and summarize it in one word, what might you call it? If it wasn't periodization, what is it? Megan, do you have any thoughts?
Like thoughts on another word?
Yeah, on another word.
The first thing that came in my head was cycling, but then I thought that's too similar to a sport. Cycling.
No,
I don't know if I can think of another name besides that. I'd have to
what is it that we're cycling, though? What are we cycling?
Loads, intensities.
Okay. Loads and intensities. Okay, good. Ryan, what about you? I forgot about you and Callie. Y'all weren't showing up on my little kind of side window here. I forgot y'all were there.
I was actually like the same thing as I was thinking of it as like a cycle.
Okay. I know why you're thinking of it as a cycle.
That was just kind of repeating, and you change it every month depending on.
Okay, you said the word in there. Anybody know what it is? Change. Okay. Periodization is change, right? Whether it is weekly changes in these ideas of called, so you talk about periodization, you break training down into what are called macro and meso and micro cycles, right? Then all that we're going to do with periodization is basically we're going to change up what it is that you're doing. You're going to do something for a day or a week or a couple of weeks or a month, and then we're going to change it, and you're going to do something else. And then we're going to change it again, and you're going to do something else, okay? In my mind, and I am in no way, shape, or form a practitioner, and Dr. Campbell would probably yell at me and tell me that whatever I'm saying is wrong. I think this is arguably the single most overblown, overemphasized thing in arguably all of resistance training is this, okay? This is, I mean, how hard is it? I did something and it's getting easier, I'm going to change it, right? Periodization is muscle confusion from the old P90X stuff. They still have P90X? You used to always see the commercials for those. I never see them anymore, okay? Muscle confusion is periodization. Muscle confusion, it's just overload. If you change what you're doing, then you're going to provide yourself a new and specific overload stimulus, right? So you're never going to get used to whatever you're doing, and so you're going to try to maximize your adaptations by changing things around, okay? Now, is there some utility to doing periodization If I'm training for a particular competition, like a powerlifting competition or something where, you know, I need to kind of work down to maximize my neural adaptations right before my competition, or I need to periodize my resistance training with my sport specific training, with my aerobic training and those kinds of things. I'm sure the answer is yes. If not, then like every strength and conditioning coach in America would be out of a job. But I also think people that try to overcomplicate this, and this is just my opinion, people that overcomplicate this are trying to sell you something. They want to sell you their way of training, okay? So that you pay them to train you, or you hire them to train your basketball team, or whatever else it is. There are good ways to do some of this. There are bad ways to do some of this. There's an immense amount of research on should you change every day, and should you change every week, and should you increase what you're doing, as you see here. Should you increase for a couple of weeks and then go back down and then increase again? And how much should you increase? Or should you start high and then taper down? Or should you do high one day a week and then low the next day? Or should you do high and then super low and then back to the middle? This is called linear periodization. What you see here is actually called reverse linear periodization, where it starts off high and you reverse the thing. So it's loading less every day. This is weekly reverse linear. You could do this one, which is like weekly reverse linear. This is sort of positive linear for a couple of weeks, and then you reset. People do this and they study this, and there's something called undulating where it's up and then it's down and then it's up and then it's down. A lot of it is bullshit, okay? Yes, there are papers that found that weekly, daily undulating over this much is slightly better than reverse linear things. There are studies that have done this. Most of the effects, to me, are not big enough to account for actual differences in inter-individual variation. Okay, there's a lot of variation in these things. They're not that much better. There's not one of them that is appreciably better than the others. The biggest mistake that people make with their resistance training is they don't overload enough, or they overload too much. Okay, they don't lift a heavy enough weight or do enough reps to get a proper stimulus, or they do the same thing over and over and over again and it ceases to become a stimulus, or they kill it one day a week and they try to kill it like 3 days in a week and they don't give themselves enough time to actually recover. Those are my kind of thoughts on things. But I am but a lowly scientist, and we all know that the practitioners, right, Keldon, don't like us scientists because we've never trained an athlete. They don't like that. So that's me. Okay. General take home from what we see from human data: how should you train? Large muscle groups, multiple exercises, probably reasonably heavy weights, probably reasonably higher volume. Oh, the short rest intervals. Did I not show the freaking, oh yeah, that data? Oh yeah, it's back here. Okay, sorry. I put in here short rest intervals. We'll talk about that next time. That used to be a thing that people from the NSCA like to promote. I don't know that we have a lot of evidence that that actually is the way to do it now. Vary your training protocol to engage different muscle fibers, allow sufficient recovery, reach some kind of firm metabolic endpoint. Nutrition is probably also pretty freaking important. So, yeah. Okay. What do we know? We know a little bit. We don't know a huge amount. Okay. Go in the weight room, lift something that seems hard, lift it until you can't lift it anymore. Do that for a couple of sets for several exercises. Come back in a couple of days, do it again. When it gets a little, whatever you've been doing gets easier, make it harder. That's it. That's resistance training in a nutshell. Same thing with running or biking. When it gets easy, go a little bit farther, go a little bit faster. We overcomplicate all of this shit. Okay. All right. Thus endeth the lesson for today. All right, I'm gonna stop sharing.